Okay, good evening. So I'd like to thank you for giving me the opportunity to discuss with you and show you the fruit of many years of research, starting with the target, topoisomerase 1, and then moving along to the development of drugs that are now in clinical development, and then the next phase, which is to move along on the how to improve this using further inhibitors of the repair pathways that the cells use to eliminate those lesions, those DNA damages. So to set the stage, I have to tell you that the targets that I'm referring to is DNA topoisomerase 1. So topoisomerases are DNA enzymes that change the topology of DNA. If you compare the DNA to a telephone cord or to a garden hose, you immediately understand how easy it is for that cord to become tangled and untangled and untwisted and totally difficult to manage. But the cells have to always divide this in two during replication and when they duplicate. And when they transcribe, they have to open up the two strands and to faithfully copy that DNA to make either RNA or DNA. So topoisomerases are essential to keep the topology of DNA coupled with the processing of all the metabolism of DNA. And that's why the cells, human cells, have 6 enzymes, in fact. And Topo1 is only one of 6. So in these enzymes, they are classified as 3 groups that we referred as Topo1, Topo2, and Topo3. The numbering is just historical. Topo1 was discovered the first, called Topo1, then Topo2, then Topo3. These enzymes, if you look at the DNA like this ladder representation, all work in a similar way. What they do, they cleave one of the DNA strands, and that enables the DNA to change its topology. You could just swivel around, or you could pass the strand through, like you would do with a garden hose. You would just clip, and then you glue back. So the enzymes are basically a pair of scissors and a tube of glue. And out of this, they could do all the magic that is needed for the cells to do their job in processing DNA. At the same time, nature has used a number of toxins that poison topoisomerases in the parasites, for instance, that invade them, and thereby kill these parasites. And that's how we found a number of topoisomerase inhibitors. They initially derived from natural product. So for topoisomerase 1, the inhibitor is Camptothecin, and for the topoisomerase 2, it's derived from a fungi, which is doxorubicin, and then from the plant, which is the etoposide derivatives. So they are specific inhibitors of both Topo2 and Topo1. So there are 2 Topo1s, there are 2 Topo2s, and there are 2 Topo3s. That makes 6. At this time, there is no inhibitor of Topo3. So I will just talk about Topo1 and about Camptothecin, and then the non-Camptothecin inhibitors that we have developed. So what about Topo1? Topo1 was discovered by Champoux and Dulbecco. You all know Dulbecco from the Medium, but one of his students was Jim Champoux. So Jim Champoux, about 30 years ago, discovered an activity in rat extracts that he called the DNA untwisting enzyme. And he called it untwisting because when he was taking any plasmid DNA or viral DNA, a drop of extract from the rat cells was enough to fully relax that DNA. And this magical activity was referred to as DNA untwisting activity. Later on, it was called topoisomerase when more of them were discovered, and Topo1 because it was the first. The way these enzymes work, so they will, for Topo1, it will introduce in one of the strands a nick in the DNA. So the enzyme would wrap around the DNA, so we'll approach the DNA, wrap around, nick one of the strands, and it then is attached covalently to one end, and the other end is free to swivel around the intact strand. And when the DNA is relaxed, meaning there is no more supercoiling, no more supertension, the DNA is realigned and is religated, and the topoisomerase goes away. And the magic of the reaction is its reaction doesn't need ATP. It works at 0 degrees. It's a very efficient enzyme, very processive. It was soon discovered that when the gene was identified in vertebrates, that the enzyme is essential. It is essential. If you knock it out, the embryo does not develop. Even in flies, the embryo does not develop. Only in yeast is somewhat dispensable. Yeast can actually use other topoisomerases, but higher organisms do not. It is essential because, as I said before, transcription and replication, as they proceed, if you imagine that you take a rope and you try to separate the strands, you would see that you have a lot of supercoiling accumulating in front. If you cannot take that out, you will not be able to open up. So Topo1 is absolutely essential. It's coupled with transcription and replication and potentially repair. Then the NCI in the 1960s had a big program for discoveries of inhibitors of anti-cancer drugs. And out of this came from the bark of a Chinese tree, Camptothecin acuminata is the name of the tree, Camptothecin acuminata decne, an activity which was very powerful as anti-cancer agent. And the chemist identified the active compound, which is this, which then they referred to as Camptothecin, just based because it was from the tree, Camptothecin. And very early on in the 70s, the drug was put into clinical trial. And the clinical trial was done with the carboxylate form, which is the form that readily opens at normal pH. In the 70s, the drug was found to be active against cancer, but discontinued because it was too toxic and nobody knew how it worked. The man who discovered this is Monroe Wall and his colleague Mansukh Wani. Monroe is now dead, Mansukh Wani is still alive, and they also discovered Taxol, so the same two men. In the 1990s, the Camptothecin mechanism of action was discovered serendipitously. SmithKline had a program to find new topoisomerase 2 inhibitors and thought that Camptothecin was a candidate. And in control experiment, they actually discovered that Topo1 was the target of Camptothecin. That changed everything. SmithKline made a water-soluble derivative of Camptothecin, which they called Icamptin or topotecan, just by putting this dimethyl amino group. So this is water-soluble. And then the drug was put into clinical trials, found to be active. At the same time, the Japanese developed a prodrug, water-soluble CPT11, irinotecan, also found to be active. And these two drugs have been approved by the FDA in about 10 years ago. Topotecan is used for ovarian cancer. Irinotecan is used for colorectal cancer and lung cancer. And both are also used for pediatric tumors. So this is about 10 years ago when this drug had been approved. The mechanism of action of these drugs remained mysterious for a long time. It was notable that the structure looks like a DNA base pair. If you look at it, it has the same shape as a fused DNA base pair, like many intercalators would do. But in fact, it turned out that the drug alone does not bind DNA and doesn't bind Topo1 alone. So in the 1990s, we in the lab sequenced a number of sites where Topo1 was trapped when it was cleaving DNA, trapped in the presence of Camptothecin. Because normally when Topo1 binds to DNA, it cleaves DNA, lets the DNA swivel, and then the DNA religates. And usually it religates a lot faster than it cleaves, so you very seldom see any of this. When you put Camptothecin, you see a lot of these cleavage sites. So I was able to sequence a lot of these sites and discovered that the bases that flank the cleavage site were selectively enriched for a thymine at the -1 position, guanine at the +1 position. And the hypothesis we made in the 90s was that the drug was binding at the interface of Topo1 and DNA, and thereby preventing the religation like a foot in the door. The DNA cannot religate because the drug is in the way. It took about 10 years to get the co-crystal structure of topotecan with Topo1 and with DNA. And this is the co-crystal structure that was obtained by Jim Shampoo, not by us, but the result is exactly in agreement with our hypothesis. This is the surface of Topo1 in gray, the DNA is green, and you could see the drug has found its way deep inside, and that's where it is localized. When you take the surface away of topo1, now you could see better where the drug is. This is the broken DNA. The broken strand is down here, and the drug is exactly wedged into the cleavage site. And this binding is reversible. The drug is not covalently bound. It just sneaks in there and just gets stuck in the topo1 DNA complex. At about the time the mechanism of action was discovered, very quickly we realized that there was probably value in finding other topo1 inhibitors. Why? Because topo1 was de facto a validated target since we knew the camptothecins were active as anti-cancer agents. Because many targets have generally multiple drugs that can target them, and the outcome is very different for different drugs. So many agents that have a common target have a very different pharmacological activity and toxicology, very different anti-cancer activity. Just take the example in the tubulin inhibitors, colchicine and vinblastine. They both target tubulin, but colchicine is used for gout and vinblastine is used for leukemia. These drugs target the same protein, but their activity is very different. So the argument was that if we found another topo1 inhibitor, it probably would target cancer differently. The other more scientific reason, or more rational, I would say, mechanistic, let's say, is that camptothecins have limitations. They are very toxic, as I said at the beginning, and it's only because medical doctors now are very good at dealing with these toxicities that they can use these drugs. They also have an intestinal toxicity, the acute diarrhea for irinotecan. They are substrate for drug efflux pumps, and therefore the cancer cells are able to take the drugs away, and they also chemically unstable, the camptothecin, and I've shown you this. What this refers to is the fact that the natural camptothecin is this structure which is called the lactone form. Under normal conditions, the lactone form is converted to the ring-open form carboxylate. In the blood of a cancer patient, when this drug is given, within 15 minutes, a large fraction, more than 50%, is converted. And this form then binds to serum albumin and is sequestered. So the fraction of the active form is always very limited. And it's hard to predict exactly how much is in the active form. So for therapeutic purpose, this is not optimum. The other limitation is the camptothecins bind to the topo1 DNA complex very reversibly. And as soon as you take the drug away, the topo1 cleavage complex is reversed. So it's good to do experiment, but in patient it means that the drug had to be given in long infusion, which is not very practical. So that was the second limitation, is there what we say pharmacokinetics is not favorable. It's a good toxin, but it's not very easy to use clinically, in spite of the fact that they are used. So at this stage, at this time, there are three classes of drugs that are non-camptothecin topo1 inhibitors. The first class was discovered in Japan. It's from natural product, from microorganisms. These are endocarbazoles, and at this time, they have not been pursued. But the two other families are totally synthetic, and one is the one we discovered, which is called the andenoisoquinoline, and the other one was discovered by Leroy Liu and Lavoie, and this one is developed by Genzyme, and this one is developed by the NCI. They're both in clinical trial, both families. So let me describe a little better the activity of the andenoisoquinoline and how the camptothecins actually bind and the andenoisoquinoline to the topo1 DNA complex. This was done in collaboration with crystallographers, and we obtained a co-crystal structure, let's say, for instance, for the andenoisoquinoline, which are the non-camptothecin topo1 inhibitor. You could see, like for the camptothecin, the topo1 now is shown in yellow, the DNA is in blue, the drug is in green. The break in the DNA is shown here at the top. The end of the DNA covalently bound to topo1 is through a tyrosine, which is shown in reddish here. And you could see that the drug fits right into the cleavage site. So it stacks between the base pairs and thereby prevents the DNA from being religated because the drug is in the way. Same mechanism as camptothecin. And by this approach, we have been able to resolve the co-crystal structure of all the Topo1 inhibitors so far, including the natural Camptothecin, which had not been done before, where the principle is the same. This is a side view like the one you have here, and then you flip it 90 degrees, which will show you better how much the drug overlaps with the base pair that flank the drug binding. So you could see here for Camptothecin, for instance, side view, the drug is wedged between the base pair. This is the cleavage site. You turn 90 degrees. This is a perfect sandwich. The base pair on the top is visible. The one under is not. The drug is exactly fitting, and by pi-pi interaction is stuck into the DNA. And that's true for topotecan. That's true for the adenoisoquinoline. That's true for the androlocarbazole. It's always the same mechanism. But it's even more interesting, because not only the drugs are held in place by the DNA, but they're also held in place by a network of hydrogen bonds. And this network, if you look at Camptothecin, is made of 3 amino acid residues from Topo1 that form hydrogen bond with the drug. One of these residue of the Topo1 form actually an hydrogen bond directly with the 20S, which is the natural Camptothecin. Natural Camptothecin, there is only one isomer which is active, which is the hydroxyl above the plan. And this hydroxyl makes a contact with this residue. And there are 3 hydrogen bonds. And if you mutate any of these hydrogen bonds, the Topo1 becomes totally resistant to the drug. The drug still binds to DNA in the complex, but it's not stable enough, and the enzyme becomes totally resistant. For the adenoisoquinoline, we have the same principle, except we have only 2 hydrogen bonds instead of the 3 for Camptothecin. So that led to the revelation to me several years ago when I went to a meeting and I gave a talk on this topic, along with someone else who was giving a talk on natural product called breferdinate, that this was very peculiar. And the person who gave the talk on breferdinate is Jacqueline Cherfis, and she had solved the crystal structure of breferdinate with the small GTPase that was targeted by that natural product. And we came with the realization that what we were finding was what we referred to as the interfacial inhibition that affects macromolecular interaction. And we named this Nature's Paradigm for Drug Discovery. So that was published now several years ago. But I think it's a concept that is quite new, and I think it's great potential. So what is this interfacial inhibition that was revealed by Topo1 inhibitors? Oh, first of all, I want to tell you how specific the Camptothecins are. These drugs, it's been remaining a dilemma in the natural product world, is how could so many plants produce the toxin which is toxic to the plant itself? Because the plants have Topo1, and the Topo1 from the plant is sensitive to Camptothecin. So initially, we all thought that it's because the drug is made in the bark, and therefore just the bark is okay. It could be toxic. And then it would kill the insects that would attack the bark. But we well knew that Camptothecin was also in the leaves. And one day I received an email from Japan not long ago. This paper was published in PNAS, where the Japanese group had actually sequenced all the trees that produce Camptothecin and had found that all these trees have a mutation in Topo1 on this residue 7-2 to serine. And that same mutation we had discovered years before in a human leukemia cell line that we selected for resistance to Camptothecin that had exactly the same mutation. And when that mutation is in place, the cell line is totally immune to the drug, so do the plant. So the reason the Camptothecin plant can produce Camptothecin is because they have a mutant Topo1, and it tells you that the drug is totally specific for that target. So the notion of interfacial inhibitors derived from the Camptothecin, and we define them as drugs that bind to a stereospecific site at the interface of macromolecular complexes, as these complexes do something. It's not a passive process. In this case, it's when the Topo1 cleaves DNA, the drug falls in, and the thing is stuck. So the drug binds to the hotspot, traps the complex, and the inhibition is generally reversible. But since the Topo1 has to work very quickly, if you slow it down, that is very damaging for the cell because then the replication and transcription will collide, and then it would kill the cells. And then I looked in, when we wrote the first review, at the co-crystal structure of many natural products, including Taxol, colchicine, alpha-amanitin, cyclosporin, ripamycin, and found the concept is the same for many natural products. The microtubule inhibitors follow the same principle. In the case of the colchicine or vinblastine, what they do is they bind between two tubulin units, and they twist the structure, and it curves it. And then the polymerization goes wrong after that. But the principle is not competitive. It just locks the system in an inactive conformation. I think this has profound implication for drug discovery because we should really pay more attention to uncompetitive inhibitors and develop assays that are based on stabilization rather than inhibition of macromolecular complexes. Today, as you hear many topics on drug discovery on kinase inhibitor, people tend to do ATP competitive inhibitor. This is not what nature usually does. Nature usually uses this type of paradigm to poison the cells and make natural toxins. So how did we go from the camptothecin to the non-camptothecin? This is thanks to a collaboration with a chemist at Purdue University, Mark Cushman. How did we get there? So in 1996, maybe, I used the NCI database that some of you may know. The National Cancer Institute uses 60 cell lines and has given this to the community where the chemists could send any drug to this database for testing activity. And many chemists have deposited these drugs. The activities are stored, and there are thousands of compounds and thousands of data points. So in the late 1990s, I thought we could use this to look for compounds that looked like camptothecin, but were not camptothecins. And within a week, Ken Paul came back to Building 37 at the NIH and said, Yves, I think I found something. And he said, I found NSC 314622. I said, what is this? He said, well, it's a compound that was made by Mark Cushman. And he deposited a few years ago. So then we talked to Mark and asked Mark, but what is it? And he said, well, it's a compound I made. I was trying to make nitidine, which is a natural product. And actually, I came up with that. And I didn't know what to do with it. So I sent it to the NCI. And it stayed there until we found it. And then Mark said, well, that's fine. It's easy to make. I'll make a lot, and we'll see if we could do better. So 10 years later, after about 500 compounds, selecting and so on, we ended up with 2 leads. And these are the 2 that are now in clinical trial, which we refer still as NSC number 776998. This has been patented by the NCI and Purdue University. Immediately it was obvious that we had already overcome one of the limitations of camptothecin. There is no alpha-hydroxylactone. These compounds are rock solid, and they work right away, and they're not metabolized very quickly like camptothecin. So that was a great advantage. So we went on. So the characteristic as far as topoisomerase 1, the way we study topoisomerase 1 in our lab, we have recombinant protein. And when topo1 is poisoned by a drug, it translates into a DNA break. So if we end-label a piece of DNA like this one, when you have a break, it makes little fragments. And you could see the little fragments generated by camptothecin here. In the adenoisoquinoline, we do as well as camptothecin, but for those who can notice, the position of the bands is different between this class of drug and the camptothecin. It means that the adenoisoquinolines are potent topo1 inhibitor, and they cleave DNA. They induce the cleavage of DNA at different sites from camptothecin, which to us was good because it meant they were not exactly identical. The mechanism was similar, but the sites where DNA would be targeted would be different. Second characteristic, I told you that the camptothecins are very transient in their inhibition. And I'm not going to describe how we measure this. But when we treat cells with camptothecin, the beauty is that as soon as you put camptothecin to the cell culture, topo1 is poisoned. As soon as you take away camptothecin, be 15 or 30 minutes or 1 hour, topo1 is released. And then the cells may go on and actually survive because it needs long exposure. For the adenoisoquinoline, what we found is even a short exposure leads to a long-lasting topo1 inhibition. So this is the experimental evidence, but you just read the title, that's enough. So the cleavage complexes persist, so that's a pharmacokinetics advantage. Another advantage is the camptothecin, as I mentioned, are substrate for the drug efflux pump. One of these drug efflux pump is the ABCG2, also otherwise called the breast cancer-related export protein. And when you overexpress ABCG2, you observe the resistance to topotecan or inotecan, the camptothecin clinically used, that are about 10 or 50-fold. It's very substantial. But for the adenoisoquinoline, we have very limited cross-resistance. And for this one, we actually have none. So again, that's another thing that we have been able to overcome. It was not that we designed the drug to do this, but it turned out that they do it. So that's an advantage. Now, the anti-tumor activity, if the drugs are in the clinic, obviously they must have some anti-tumor activity in animal models. So this was done, and really it's because of the NCI support, the DCTD support, and Center for Cancer Research that this was feasible, because once you go into animal, you're dealing with very complex experiment and high cost. So this was possible. And when we were doing this, what we realized, since we wanted to put this drug in clinical trial, nowadays it's very difficult to do a clinical trial without having a pharmacodynamic biomarker, meaning something that you could measure during the first administration that will tell you that the drug is really doing something. Because usually in phase 1, all you do is look for toxicity. You're looking for the maximum tolerated dose. But nowadays you want more. In phase 1, you already want to have a proof of principle that you are having some kind of activity, and you want a pharmacodynamic molecular biomarker. So we were lucky because in the lab, in the molecular pharmacology lab, is Bill Bonner, William Bonner. And he had discovered several years before that during DNA damage, chromatin responds by phosphorylating one histone, which is histone gamma H2AX, histone H2AX. And this phosphorylation happens when DNA is undergoing DNA double-strand break. And we knew that with camptothecin, there is formation of DNA double-strand break. And this is what kills the cells. So in the case of camptothecin, the topo1 normally binds DNA, and topo1 religates, and usually it religates faster than it cleaves. So this is almost undetectable. When you add camptothecin, you stabilize, so you force the equilibrium this way. But this is not really lethal unless replication comes in. Once the topo1 is stuck, it's like on 270. You know, if you have a truck, just stop in the middle, and you go 50 miles an hour. If you don't have the brakes, and if you're just going to collide. And that's what kills the cells. And during the collision, the break, which is initially a single-strand break, is converted to a double-strand end by the leading polymerase collision. And this is enough to activate the gamma H2AX. So we knew this. So I turned to Bill Bonner. This is William Bonner. So for those who would come to Building 37, this is him. And he discovered not only gamma H2AX, but he discovered the gene, which he called H2AX, and then discovered the phosphorylation, made the first antibody, and it's thanks to Bill Bonner that this is used by so many people now in the world. So we took advantage because Bill is the next door neighbor to me. And so gamma H2AX forms, if you take the chromatin fiber, there are about 10 to 20% of the nucleosomes contain one H2AX. It's randomly distributed. And the H2AX histone is characterized by an extension of the tail that has a serine residue which is phosphorylated by ATM or DNA PK in response to double-strand breaks. And this can then be detected by antibodies. So when a double-strand break is formed by radiation or by replication, the kinase, ATM, ATR, DNA PK are activated, phosphorylate H2AX, forms gamma H2AX, which then recruits a number of proteins to chromatin, DNA repair protein, remodeling protein, cohesin, and then this favors DNA repair. If the DNA repair is efficient, the cells survive. If the DNA repair fails or is insufficient, then there is even further gamma H2AX and the cells die. This is the way it looks. So if you take a normal human lymphocytes, usually you have almost no background, sometimes one gamma H2AX focus. The DNA here is that the nucleus is this red PI stain image. And when you treat with camptothecin, you could see all these gamma H2AX foci. Each focus is one double-strand break, which is remarkable. You could detect one single strand break. And you could do this not only in cells, treated cells, but you could do it as well in the tumor needle biopsy of treated patients. This is the tumor without treatment. This is with treatment. And you could detect it in the hair, in the plucked hair of a cancer patient treated. Then the normal hair has very little signal. In the cancer-treated patient, you can see a signal because the hair follicles have many replicating cells. Many drugs are known to activate gamma H2AX. In fact, many drugs that target chromatin. And camptothecins were the first to be described. We described this in 2003 with Bill Bonner and showed that at very low dose, pharmacological concentration and short time, gamma H2AX is activated, easily detected by immunofluorescence microscopy. So now we went back and we say, okay, could we use this and could we do it in the context of mice? First, what we wanted to do is to make sure that the reference compound, topotecan, would do it at various doses under what we call phase 0, which is phase 1 associated with biomarker. And then once we could do this, could we do this with the adenoisoquinoline to determine in the patients who are going to be treated, when should we sample the tumor? Because it's easy to say, but it's another thing to tell a patient, we're going to biopsy your tumor at 2, 4, or 6 hours, but not going to do it at the 3 times. We do it once. So you have to know when. So we used the mouse model to do this. And this was in collaboration with Jim DeRocho's group. So these are mice, for instance, tumor-bearing mice. There are 4 different tumors. These are untreated, and you could see very little background. There are a few cells that are gamma H2AX positive. If you treat with the adenoisoquinoline, you could see a lot more green foci. So these cells become positive, and this is an expanded view. So it does work at pharmacological concentration. The other thing we learned from mice, which is relatively interesting, is that you can cure mice with camptothecin. If you establish a cancer in a mouse, camptothecin can cure them. Unfortunately, you cannot cure humans with camptothecin. And as much as I like these drugs, this is not feasible. And the reason it's not is you can never reach the dose that will enable you to do this. And the reason is that mice, and to some extent children, their bone marrow is a lot more resistant to chemotherapy. And you could then double or triple the dose. It is just enough to achieve a full response. And so what we learn from the mice is if you compare a human versus a murine bone marrow progenitor, the human bone marrow progenitor is 10-fold more sensitive than the murine one. And this is the reason you could cure mice and you cannot cure humans with camptothecin. So when we did the adenoisoquinoline, we're curious to see how this translated for the adenos. And the situation is a little better. We're almost the same, let's say half. So the human cells are only half more sensitive, meaning that if we get response in mice, it's more likely it will be meaningful in the context of human tumors. So this is the activity in mice of the adenoisoquinoline. So this is one tumor grown in mice measuring the volume. And you could see without treatment the growth of the tumor and the tumor volume increasing. If you treat with topotecan at the highest tolerated dose, which is this curve, at this dose, the mice lose about a quarter of their weight, and they're almost moribund. So often they would have to be sacrificed anyway. In the adenoisoquinoline, at doses that are much less toxic, we get a much better response. So this was a fairly optimistic result. And at these conditions, when we get these responses, we measured gamma H2AX. And under these conditions, at about, this is maximal tolerated dose, the abbreviation, at about half of the maximal tolerated dose of mice, so half of what it would take to really make the mouse very sick, we get a very strong gamma H2AX signal. And we also determine the time course, and the optimum is 4 hours after a single administration. So it told us that in patients now, we were ready for the clinical trial to say that in the patients, after the single administration, we should do the biopsy at between 4 and 6 hours. Because in the clinic, you cannot be that precise. So you'll go 4 or 6, but we'll know when, but it's not always exactly the same time. So we were ready to go, and now we are in clinical trials. So in Building 10 at the NIH, patients are being treated with these 2 adenoisoquinoline. So the adenoisoquinoline are potent. They are specific topo-1 inhibitors. They are chemically stable, whereas the camptothecins ring open. They induce topo cleavage complexes at different genomic sites, which suggests that they could probably produce different anti-cancer activity. They induce topo-1 cleavage complexes that persist after drug removal, meaning we could just give 1 dose and that would last for a long time. They overcome the drug efflux resistance, which is otherwise a problem with the camptothecins. They are anti-tumor. They are less toxic. We can use histone gamma H2X as a sensitive biomarker, and then this is now in clinical trial. So we're still in phase 1, and it takes time because for each patient, we have to get the patient who are agreeable to get the tumor biopsies, which is very unusual. Usually in phase 1, you do not get tumor biopsies, but patients are very forthcoming, and actually they agree to do this for their good and for the good of everyone. So this is ongoing now. So I'd like to finish the next few minutes to tell you the next stage of where we see this going. The topo-1 DNA complexes that are formed in cells, the cells actually have ways to deal with them. And there is a special repair pathway that removes these cleavage complexes. The name of this enzyme that removes topo-1, and I'm in part responsible for the name, but it's not. Howard Nash at the NIH discovered this enzyme from yeast initially. He found an activity in yeast extract that was able to remove these topo-1 DNA complexes. We talked with Howard and came up with the simplest possible accurate name, which turned out to be tyrosine DNA phosphodiesterase. The reason it's called that is because the amino acid that is linked to DNA in the case of topo is a tyrosine. And what the enzyme does, it cleaves between the tyrosine and the DNA, the phosphodiester bond, and that's called DNA phosphodiesterase. So it's called TDP1. So the way the enzyme works, when topo-1 is stuck at the end of the DNA, like here, it's a large protein. It's about 100 kilodalton. There's nothing that could really remove this very easily. It's a very large, bulky adulton DNA. To repair the DNA, this needs to be taken off. And this enzyme TDP1 will remove this. There are other alternative pathways, but I don't want to deal with this now, and it will take that off. So there is a good knowledge of this enzyme. It exists as a recombinant protein. There is crystal structure. We know it functions. So this is the crystal structure of this TDP1. It's about 70 kilodalton protein. It almost looks like a dimer, but it's not. It's a monomer. And it has these active site residues here, which are called these HKN or HKD motifs. HKN motifs, there are 2 motifs, and they come together like a pair of tweezers, and they get around the DNA. So the DNA gets into a groove, and then the peptide is here, and the phosphodiesterase activity cleaves the peptide. Several years ago, it was a serendipitous discovery. There is a disease in humans which is called a neurological disease, fairly rare, which is called spinocerebellar ataxia, which is called SCAN. And when the people sequenced the genes that was responsible for this disease, they found that it was TDP1, tyrosine DNA phosphodiesterase. It was a total surprise. But it meant that actually this repair activity was essential in neurons to prevent or remove topo-1 under normal conditions, and maybe that was a very essential function. So this disease, SCAN1, is a homozygote mutation of the TDP1 gene found initially in a family in Saudi Arabia. And this is this mutation, and the amino acid that is being mutated is this histidine 493 to arginine. The mechanism, for those, and I'll go very quickly through this, is normally when topo-1 is bound through its tyrosine here to the end of the DNA, so this is 3' end of DNA. The way the enzyme works is it approaches this, so binds to the topo-1 DNA complex, and works in 2 steps. In the first step, the enzyme serves as makes a covalent bond
to the DNA, to the phosphate, which enables the unlinking of the tyrosine, so it frees the topo1. But there needs to be a second step to remove the bond between tyrosine DNA phosphodiesterase and DNA. And this requires this amino acid, histidine 493, to activate the water and then dislodge this bond and then free the DNA. So it's a two-step mechanism. And beautifully enough, the mechanism of the scan mutation is mutation of this residue 493. So 493 is mutated to arginine, and arginine is not able to activate the water, and the enzyme TDP1 gets stuck on the DNA. So instead of removing topo1 and giving back everything normal, there's like a switch. You switch from a topo1 DNA complex to a DNA TDP1 complex, and that's very toxic. So what happens is then these scan patients are unable to remove topo1 DNA complexes, and they form TDP1 DNA complexes. So we looked at these scan cells from patients, lymphoblastoid cells, and these are the cells from the scan patients in circle, and these are in squares, and the control cells in circle. And this is survival, and you could see that the cells that are in square are more sensitive than the cells in circle. So the scan DNA repair deficient cells are more sensitive to camptothecin. And this is the removal of topo1 DNA complexes. The normal cells remove the topo1 DNA complexes a lot faster than the scan cells. But these cells are not very easy to use. So we generated recently an isogenic TDP1 deficient cells to screen for TDP1 inhibitors. Because our rationale that now that we have topo1 inhibitors, if we also have TDP1 inhibitors and we give them together, then we should enhance the activity of camptothecin. And this work, which is ongoing, is done in collaboration with researchers at Kyoto University. Junko Mura is in our lab, and Dr. Takeda is in Japan. So what we did, we took advantage of the fact that in chicken, there are chicken cells called DT40 cells that are very highly recombinogenic. They recombine very easily. That's their intrinsic characteristic. And they are very easy, relatively easy, to make knockout. So we can make isogenic cells very quickly. And those cells grow very quickly. They have 8-hour doubling time, almost like yeast. So it's very good for screening. And we've just been successful at generating genetically inactivated TDP1 cells. And this is just the classical way we do these things by recombination. And so those cells we have generated actually have normal doubling time. They don't mind not having TDP1. Actually, the patients who are deficient in TDP1 live a normal life until early adolescence, and then they have very severe neurological disease. But in cell line, it doesn't really matter. They double fine. But when we look at the TDP1 activity, and the way we could do this is we could take small oligonucleotides that are labeled, and then we have a tyrosine at the end. And if you have TDP1 activity, you will remove the tyrosine to a phosphate, and the DNA, if you migrate it on a gel, so this is the regular big DNA with a tyrosine, when it's cleaved, then it migrates faster. And in a normal cell lysate, you could see as you increase the amount of lysate, the conversion of the starting material and removal of tyrosine giving the product. In the TDP1 deficient cell, there is a total inactivation, meaning there is no alternative enzyme. This is the only enzyme, at least in chicken cell, that can do this. So this is a very important enzyme. This is the quantitation. And those DT40 cells, when we test their sensitivity to camptothecin, you could see it here. The TDP1 deficient cells are very sensitive. The wild-type cells, shown in circle, at concentrations of camptothecin that are very much subtoxic, low nanomolar. Under this condition, the normal cells are almost totally resistant. They don't see the drug. But those TDP1 deficient cells, 99% of the cells can die. When we retransfect the knockout with human TDP1, we restore the resistance. So now what we have been able to do is that we have been able to put a grant. So near here you have the NCGC. You probably have seen the building. So the National Chemical Genomics Center has a huge screening program. And if you apply for a grant, which we did, you could then have them screen with you for their libraries of compound to look for inhibitors. So we put in a grant, which we call an RS3, which has been submitted to the NCGC, to screen for TDP1 inhibitors. We're the first, I think no other labs or group has ever done it yet, for compounds that would synergize the cell killing of camptothecin selectively in the TDP1 knockout cells, complemented with human TDP1. So if you have a drug that synergizes only when you have TDP1, it means a TDP1 inhibitor. If it synergizes independently of TDP1, it's a non-specific DNA repair inhibitor. So we have a system now to do this. And then this can be put in the context of other screenings that we have also already developed. So we have high throughput biochemical screen that are already going with recombinant human TDP1. So we have 3 screens that are ongoing. One in our lab, the Laboratory of Molecular Pharmacology, with this Dr. Christophe Marchand. Another screen with the Molecular Target Laboratory with Dr. Barry O'Keefe, who is here, who has set up the screen in his lab for natural products. And then the screen at the NCGC for all the library of the NCGC. So anything could be put in the context of this bigger enterprise. So in the end, I'd like to acknowledge, especially for the drug development part, because it's, in fact, it's a very unique opportunity to have a drug go from the lab to the clinic. I think the fraction is very small. It's probably in the 10 minus 2, 10 minus 3, maybe 10 minus 4. It's also very, probability that a drug in phase 1 makes it to commercialization is also very low. I'm aware of this. But we've been very fortunate to be able to go at least from lab to patients. And this is really thanks to Mark Cushman, who is the chemist at Purdue University. Initially, Ken Paul with disease now, but Ken really helped me through the DTP and the NCI 60. And then Jim Doroshow is the director of the Division of Cancer Treatment and Diagnosis, who is also a PI in the LMP. So I'd be happy to entertain any question if you have any now or after the talk. So thank you for your attention.